Hi, this is Eric Lyons, and this is a video tutorial showing our new SNP visualization system, which is part of the EPIC-CoGe Browser. I'm going to start out by logging in with CoGe's test user account. If you have a CoGe account, you're welcome to log in with it. You can, of course, use the system anonymously. All the data that I'm going to show is fully public. And what I've done is I've have a notebook created for the CoGe user that has all of the SNP data that we've currently loaded into it, which is under this folder, Arabidopsis SNPs. So here's a list of all of the initial sets of SNP data. These all come from the 1001 Arabidopsis Genome Project, and we've loaded in 343 of those data sets. So for just taking a look at any one of these, we can bring up what we call the experiment view page. This basically tracks all the metadata. So this particular experiment, again, is from the 1001 Genomes Project, and specifically from the project MPIC Wing 2013. We've got links in the metadata system that allow you to go take a look at this particular set of data. Here's the original project page, including links to download all of the raw data. It includes a bunch of stuff in terms of who's on the project, where it was sequenced over at the Max Planck Institute for Developmental Biology, the PIs that are involved with it, and interesting stuff like lat-long position, so we can do fun things like get a Google map link so you can actually see where this particular individual was collected. And then what you have is the opportunity to view this. And so if you view it, you bring up the EPIC-CoGe Browser. This here is the SNP track that we happen to select. I'm going to also turn on our features for this genome as well. There can be some odd rendering once in a while where those things get overlaid. I'm going to bring our feature track up to the very top. So here's our individual features. I can do the traditional gbrowse version of showing genes, mRNAs, coding sequence, all those kind of things as well. And if I go ahead and zoom in on a particular region, for example, this spot right here, we can start to see some of these individual SNPs start showing up. So here's some of our SNPs, and as I continue to zoom in, we can start to see those as individual nucleotides. So every single kind of transition or transversion has a unique color on it in terms of one of these substitutions. And as we get close at a certain resolution, we start to see details of that particular SNP. So here we have a deletion, here we have an A to T transversion, a G to T transversion as well. Along those lines. What's nice about this system is that you can easily add in and mix any other sets of data, experimental data that we have. So we've got, I think, about over 1,000 experiments that you can select from, including methylation data. So I'll select one of those, and we'll see if we've got some observations at this point. And let me go ahead and zoom out. You can start to see some of these data sets show up. But we also have expression data in the system. So here's a nice area. We'll wait for these tracks to regenerate. But here's a region that has a bunch of SNPs as well as a bunch of methylation in this particular background. I'm going to turn this track off. We can also show expression. So I'm going to look for some of the RNA-seq data. And these can occur in a variety of backgrounds. So here you can start to see expression of particular genes. And it's nice to be able to have all of these data integrated. So for example, if you're interested in this particular region, in terms of both the SNPs and the methylation pattern, you have the opportunity to quickly bring those data together, have the opportunity to zoom in, see where those SNPs are, see what the underlying methylation patterns is, and if there is any expression, you can take a look at those as well. Just to remind you, if at any point you have a particular view or set of experiments you like, you can easily generate a link so that you can revisit this in the future. If you're logged into the system, it'll remember which tracks you've selected, so you can see those again. And if you are in your user profile page, you easily can go ahead and add in new experiments to any genome that's in there. So this lets you mix and match public and private data, as well as be able to share those with other users in the system. So hope you found that useful. If you do have any questions about the EPIC-CoGe project, just do a Google search for EPIC-CoGe. You'll get the overview of it in Cogepedia. We've got a tutorial page up. There's a video on how to use it as well. Definitely visit the EPIC project, which is the Epigenomics of Plants International Consortium. And if you do have any specific questions, please feel free to email us. Just go to the help button, go to contact or cite us. If you have specific questions about one of these tools, just click on the page docs. Visit us on the forums, ask any questions that you have. Thanks a bunch.